if we just look at the obesity epidemic from a bird's eye view, and we look at the
Hmm.
spatial and temporal pattern of obesity spread over time, where it has risen faster, the types of people it's affected, which is basically everyone, and we look at its sort of rate of change over time.
Hmm.
When you think about sort of just in principle the different types of models that people have had out there for what could be causing this in principle, there's one school of thought that is more or less that a lot of it has to do with genetics per se, that a lot of obesity is inherited in the DNA sequence. There's other schools of thought that say, well, it's mostly just diet and exercise. It's how
Hmm.
much you move your body and how many calories you put into your body.
Yeah.
And then, of course, there's a third potential piece of this puzzle, which is things like obesogens, things in the environment which might be promoting obesity. If you just look at the general pattern of obesity spread over time, does it get you thinking a little bit more or a little bit less in one direction? Is there anything at a high level that can be ruled out?
I think the first thing to be ruled out is genetics, right? The obesity pandemic has happened too fast over too many large populations for there to have been a change in the underlying genetics. So I think genetics is out as a cause of the obesity pandemic. Are there mutations that can make people predisposed to become fat? Absolutely. Are they spreading through the population at warp speed? Not a chance. So the primary paradigm, and there are many people who vociferously and angrily defend this point of view, is that it's all about energy balance. It's simple matter of thermodynamics, that it's the number of calories you take in minus the number of calories you burn. And if you make that balance, that you are not going to gain weight. And that model has, I mean, at some level, it must be true, right? You can't get fat from breathing the air. You have to incorporate more of the calories you consume into your body, right? But the question is, how does that happen? So most living organisms have a homeostatic control of appetite and satiety. Most animals don't overeat. You won't go on safari and see very many fat animals. You don't see fat animals until you look at, our pets and you look at the domestic animals that we feed. So something is disrupting the homeostatic control of appetite, satiety, and weight. There's another very important model called the carbohydrate insulin model.
Mm-hmm.
It says the amount of simple carbohydrates in our diet are causing excessive stimulation of insulin, which is causing excessive storage of fat. So the carbohydrate insulin model does a pretty good job of explaining a lot of obesity because carbohydrates have gone up like crazy in our diet, but they don't do a good job of explaining the homeostatic control of weight and appetite and satiety.
Hmm.
There's the obesogen model, and there's another kind of minor model called the redox model, the energy that says that the burning, if you will, of fuel molecules, the consumption, the metabolism of fuel molecules makes a certain amount of reactive signaling molecules that affect appetite and satiety and metabolism. And then there's obesogens. And I think we don't have enough data from epidemiology studies to say to what extent obesogens are responsible for the obesity pandemic. But what we can show pretty convincingly in animals is that if you expose, in our case, mice, to very low doses of tributyltin, that they will get fat, and so will their descendants. So that to me is the most fascinating thing. How are these effects of an environmental exposure transmitted to future generations without changing the sequence of the DNA?
Hmm. So you expose mice to TBT, it promotes obesity. How much TBT are you giving them? How are you giving it to them? And exactly how obese do they get?
Exactly. We're putting it in their drinking water, right? Because that's the simplest way that people get exposed. I don't, pump it down their throat or inject it subcutaneously. We want to try and make a more realistic exposure. We're giving them 50 nanomolar, so more or less 50 parts per billion, which is around the dose at which you can find in humans. You can find that the average in some studies in human blood was 20 nanomolar.
Hmm.
So we're only giving them 50 in the drinking water. So how much are they achieving in the blood?
And how do we think humans are ingesting this? Is it in our drinking water?
No one's measuring it, I can tell you.
Hmm.
It's not a chemical that most water agencies measure. It's in many kinds of plastics. It's in the vinyl plastics in your home. TBT is used on some food crops. It's used as a miticide on nuts and stone fruits. So we don't know a lot about where the exposure could be coming from. We just know that it's out there. When you look in people, you find it. It's hard to measure. So not so many labs do a good job of measuring it. So that contributes to the uncertainty of to what extent are we exposed. And I'm not trying to say that tributyl is what's making us fat. What I'm trying to say is here's an example compound which we know is in the environment. We have some idea of the level that's in the environment. We have some idea of what levels cause effect in animals, and those effects are permanent. What it does to our mice is it makes them predisposed to gain weight when we up the fat in the diet a little bit.
Okay, so
So
they're not just automatically getting fatter when they are exposed to TBT, but when you increase the fat content of their diet somewhat, now they do.
yeah, they get a little bit fatter on a normal diet. So a normal diet, the normal mouse chow that we give them is 13% by calories fat. And at that level, if you keep them long enough, the animals whose ancestors were exposed to TBT will get a bit fatter. But if you switch them to a higher fat diet, and by high fat diet, I mean 20% of calories from fat, which is still way low fat diet. We're not giving them, 60% calories from fat, which some people do. We're giving them a relatively modest increase in fat. They get obese within a few weeks.
Hmm. I see.
And when
So it
and it's only the
predisposes them
males.
to put on the fat. There's a sex
Hmm.
specific effect. It's only in
Yes.
males. And as you said, it's quite a modest increase in the fat content. It's not like they're having
Yes.
they're not on like a ketogenic diet.
Yeah. From 13 to 20%. And even 20% is considered to be a low fat diet. But
Hmm.
compared to 13%, it's a high fat diet. But if you keep the animals on a diet that's like 18% fat and then switch that to 40% fat, it's hard to see the difference
What
because all the
happens?
animals get fat really fast.
So they get fat faster due to this
Mm-hmm.
ancestral exposure to TBT.
Mm-hmm.
So there's this transgenerational effect. What happens if the mice try and lose weight?
Well, we've never tried to do that, right? We feed them ad libitum. We just put the food there and let them eat as much as they want. Surprisingly, the animals whose ancestors were exposed to TBT eat less. They don't eat more.
Huh. So they're getting
And
fatter despite the fact that they're consuming less.
yes.
Wow.
And how much less is, it's debatable because we're doing experiments with cages of 4 animals, but at least you can say is they don't eat more, right? If they get 15% fatter, they didn't eat 15% more food. We can't detect any increase in the food, and it looks like a small decrease. And when we put them into metabolic cages, we didn't see any big changes in the respiratory activity. We didn't see that they were less active. We didn't see that they breathed out a different amount of CO2 than did the others.
So if you expose animals to TBT, they get fatter if you modestly increase the fat content of their diet.
Mm-hmm.
The same effect is observed in their offspring, even though they are not exposed to TBT. So there's an inheritance of something. What exactly is being inherited?
So that's the $64,000 question, right? What is being inherited? So if you let's discuss how we did the experiment. So we took a pregnant mouse. We actually exposed her before she was pregnant, right? We exposed the mice, females for a week to tribulletin. Then we took it away, put the males in the cage, left the males there for 3 days so that they could, do their business, and then took the males out and put the TBT back in the water. So those females, if we ever keep them to adulthood, which we don't usually do, they will get fat. But the offspring, so the offspring of those females, so in genetic terms, let's call those females F0. the filial generation. So then the offspring are the F1, and the males get fat, the females don't when we switch the diet. If we breed the animals, and not the ones that got fat, breed them before they get fat, breed their siblings to make the F2 generation, same thing happens. The males get fat, the females don't. And we've gone out as far as the F4 generation. So the great grandchildren of the exposed animal. So you can imagine, if you think about how it works in mammals, right? So the fetus is inside the mother. So the F1 generation was exposed directly to the chemical while it was in the mother's uterus.
Mm-hmm.
But the F2 generation was a germ cell inside the F1, so it's exposed also. So when exposing a pregnant female experiment, the F0, the F1, and the F2 generations are all exposed to the chemical in some way.
And if it's specific to males, I imagine there's some kind of specific effect on the sperm.
It could be, but we don't see that.
Mm.
So it seems to be coming through both germ lines.
I see.
So if in a pregnant female exposure, the F3, right, the great grandchildren is the first unexposed generation, and the F4, the great grandchildren, they're not exposed. And because we treat large amounts of animals, right, when you do these experiments, we typically will treat 25 females with vehicle and 25 with TBT. So it's difficult to imagine that we caused the same mutation in every one of those animals. That would be unreasonable to think that TBT happened as a single mutation. So I can tell you I haven't sequenced the genome of all the animals, so I can't definitively say there is no mutation, but I think the chance that there is vanishingly small.
Yeah, that makes sense. So these animals are different, clearly.
Mm-hmm.
What exactly is different about them? I would imagine that they're expressing different genes more or less than they otherwise would be if they hadn't had an ancestor exposed to TBT.
Yes. So what our belief is, our model that we've been working on since we published a paper about this in 2017, is that this exposure has caused a change in the structure of the DNA in the nucleus. So the DNA in the nucleus interacts with a lot of proteins, histones, and other proteins, and that combination is called chromatin. And it makes loops, small loops, big loops, and it's folded up in a pretty compact structure. But it turns out that parts of the genome always like to be near other parts, right? So if you can think of the genome as a bowl of spaghetti, but that spaghetti has an inherent structure, and there's parts that always want to be near other parts and want to be away from other parts, and that localization of these various loops, small loops, big loops, affects what genes are expressed. Mm-hmm. So what we believe is that the exposure to TBT or potential some other chemical has caused that three-dimensional structure to change, such that some genes are more expressed and some genes are less expressed.
Mm-hmm.
So we tested that, and we found out that when we looked at the germ cells, we're looking at the primordial germ cells, right, the before from embryos that are about 2 weeks old, we can see new loops in a particular part of the genome.
So even though we're not talking about DNA sequence changes here, the three-dimensional geometry of the genome is changing.
Yes, that's correct. And what happened is we put a new contact smack in the middle of a gene. And when we first found out this, we said, what is this gene? What does this do? And we looked at, oh, that's the gene for insulin degrading enzyme. So these animals have excessively high insulin levels, especially when you give them a higher fat diet. The insulin levels go to the moon, only in the males.